This is the lecture that goes with your lab that will require you to draw a diagram of meiosis and also mitosis. There'll also be a diagram of mitosis, but that one is comparatively simple. So maybe we'll say meiosis plus mitosis lab. So here we go. Remember that what meiosis is in human beings and mammals is the production of gametes or sex cells that have half the number of chromosomes that a somatic cell has. A somatic cell is basically all of the other cells in the body. These sex cells undergo fertilization, they fuse, which brings the chromosome number back to 2n, or diploid. And here we go. Before we begin, I'd like to review what homologous chromosomes are, because that is what is getting divided during meiosis. So what are homologous chromosomes? Number one, a chromosome, shown here, is a long condensed piece of DNA and also proteins that participate in condensing the DNA. Gene loci, that is the location of genes. So where are the genes? Here's one and there's one. These arrows happen to be pointing towards darker bands. The bands do not represent genes. Bands are the result of staining. This shown here, 1, 2, 3, 4, these four things, they are two homologous pairs of chromosomes. Two homologous pairs. Okay, so a gene. What is a gene? It is a length of DNA that codes for a particular protein or a polypeptide. So a protein could be an enzyme, it could be an antibody, it could be a active transport protein. A gene locus, plural is loci, is the specific position of a gene on a chromosome, and an allele is a particular variety of a gene. For example, the classic example, blue versus brown eyes. Two alleles of the same gene. Gene is eye color, allele is type of eye color or version of the gene. So lastly, and the most difficult one, is homologous chromosomes. They are a pair of chromosomes in a diploid cell that have the same structure as each other. What that means is they have the same genes, but not necessarily the same alleles of those genes at the same loci, and that pair together to form a tetrad during the first division of meiosis. Notice I read through these definitions. That means I really want you to know these definitions. What's a gene? What's a gene locus? An allele and homologous chromosomes. Shown here, this is a karyotype of a human male. We know it's male because there's one X chromosome and one Y chromosome. This is the whole genome. That is the whole of the genetic information in an organism, and we've got 23 pairs, including these sex cells here. Each of these is a homologous pair. There's a homologous pair, there's a homologous pair, etc., etc. All of them are homologous pairs. And in this karyotype, they have the S phase has not occurred in this cell. If S phase had occurred, what this chromosome pair would look like after S phase would be more like there's one chromosome, and then it would have its sister chromatid attached to it, and then we'd have the second chromosome also with its sister chromatid. You have to forgive me, I'm not very good at drawing on a computer. Okay, that would be, so that's after S phase. Okay, moving on. Okay, a couple of questions for you before we move on. This is shown here is a homologous pair of chromosomes. A homologous pair of chromosomes. This one came from the paternal side, or dad. This one came from the maternal side, or mom, if we're talking about human beings. So ask yourself, do they have all of the same genes? Yes or no? You can pause it. The answer is yes. They have all of the same genes. So yes, and you can think of, again, the example of eye color. Do both of your parents contribute to the determination of what eye color you have? Yes, they do. So let's take a look at maybe if this was the gene for eye color, or the locus for eye color. You can see that both the paternal and maternal homologous chromosomes here, they both have a gene there, but they are different. They are different versions of the gene, but they are in fact the same gene. And this is just a cartoon version, so it's not a perfect analogy. Next question is, yes, they're homologous chromosomes. Do they have all the same alleles? And I already kind of gave away the answer to that one. The answer is no. They do not have all of the same alleles. So, for example, perhaps this symbol here, this light colored box, maybe that codes for blue eyes. And then over here, this dark colored box, it's just a symbol. Maybe that codes for brown eyes. So both chromosomes have a gene for eye color at the same location. That is, kind of the upper part here and then the upper part there, but they have different versions of the genes. Okay, so here we go, moving on. Now we're going to cover each stage in detail, and you're going to need to know what is happening to the homologous chromosomes and sister chromatids during each phase of meiosis 1 and 2. So sister chromatids and homologous chromosomes. Make sure you know what those are. So in this diagram right here, what we are showing is the beginning of meiosis 1, and then that's going to include prophase 1, metaphase 1, anaphase 1, telophase 1. And then over here is the beginning of meiosis 2, which includes all of these phases, same ones, except we put a number 2 on it. So just to give a brief outline, at the beginning of meiosis 1, we would see prophase, the nuclear envelope is intact, and homologous chromosomes pair up, or they synapse, and this is called a tetrad. This is another tetrad. Prophase 2 of meiosis 2 does not, there are no more homologous chromosomes. Notice the second chromosome here is no longer present in this diagram. That's because division has taken place. If you're a little confused on that, well, you can either review or keep going and it should get cleared up for you. This is a link that if you'd like to access the PPT for this, you can right click on this picture and extract a hyperlink from it. If you're watching this in presentation mode, the PPT that is, then you can just click on the picture. And if it is not in presentation mode, then you can right click on it. And if you've got questions about that, you can ask me, email me. So remember that in interphase in summary, in the S phase of interphase, before meiosis starts, DNA replication occurs. So that's why we've got sister chromatids there. Chromosomes are replicated, and these copies are attached to each other at the centromere. There's the centromere. There's the centromere. That's the and each copy, they're an exact copy of each other. This is an exact copy of that result of DNA replication. While they're attached, we call them sister chromatids. So this is a pair of sister chromatids. This is a pair of sister chromatids, and this is also a pair of sister chromatids. Following S phase, further growth and preparation take place for meiosis. And that's again just going to be a checkpoint, and that would be shown down here. The G2 checkpoint, have they made all the proper preparations? So our goal here is to describe the behavior of chromosomes during meiosis, the behavior of chromosomes, which mostly means the movement of them. So we're going to have to use those terms, homologous chromosome and sister chromosomes, accurately. Meiosis in a quick summary. A homologous pair of chromosomes. During S phase, they are replicated, joined at the centromere. And after that, homologous pairs associate during prophase 1 through synapsis. That is called a tetrad. Crossing over may take place between non-sister chromatids, in which case alleles are exchanged or genes are exchanged between non-sister chromatids, and that leads to a recombination of alleles. Then they are separated. The homologous pairs are separated. That happens in anaphase 1. The homologous pairs are separated, but the sister chromatids remain attached. Notice the centromeres are still present. This is the reductive division. So one of these chromosomes is going that way, right? And that way. And they're going to go into different daughter cells. So the daughter cell on the right is going to have, and I'm going to simplify things a little bit here, it's going to have the allele that is coded for by this darker square, and the allele on the left, the cell on the left is going to have this allele of the gene. So the daughter cells will not have two copies of this gene anymore. They will have one copy of the gene. meaning so there's not two versions anymore. There used to be that version and this version, but now that we have divided them, each daughter cell will only have one of those versions. Okay, so we're going to now go through this in a little bit more detail. So prophase 1, this is when crossing over occurs, and that is the exchange of DNA between non-sister chromatids, and it happens in prophase. Only prophase 1, not 2, not in prophase 2. So if we're looking at this pair of chromosomes, and we're looking at it down here, a little zoomed in, let's give some of these genes a name. So we could say gene C, we could say gene D, and then down here, gene E. Now remember that there are things called alleles, which are different versions of genes. So this chromosome, this is a chromosome, we'll call that chromosome 2, and then we'll call this one chromosome 1. Chromosome 1, let's see, for gene C, there's a big C here, and big C there, and then chromosome 2, also big C, big C. So they have the same allele, same version of the gene. What about gene D? Gene D, well, there's two capital D's here, then there's a lowercase d here, lowercase d there. And all that is symbolizing is that they are different versions of whatever this gene is. One of them is symbolized by a capital D, and then the other one symbolized by a lowercase d. And then the same thing down here for gene E. The pink chromosome here, chromosome 1, has got these little e's, so that's one version. And notice this one's kind of been twisted around here. That's still the pink chromosome, though. And it also has a little e there. And then the blue chromosome, chromosome number 2, has got a big E here and then a big E here as well. So this is where chiasmata formation happened. This is chiasmata. This is a chiasmata. Which one's plural and which one's singular? Chiasmata. Chiasmata is plural. Sorry about that. Chiasma formation. I might have that backwards. Sorry about that. Anyway, exchange of DNA. So originally, chromosome number 2, what you would always get is you'd have a little d and a big E together. But now, since there has been an exchange of DNA, we're going to go cut. We're going to now end up with a little d and a little E, which was not a combination that was present before. We'll go into this a little more. This is a summary of what I've just said, or a similar description, and I'll let you read that if you want to on your own time. Okay, so what I was just saying there. First, during, again, this is that's not how you write it, prophase 1, first synapsis occurs, meaning homologous chromosomes associate. Chiasma formation, that's the singular. Singular is chiasma formation. Chiasma formation occur when neighboring non-sister chromatids are cut at the same point. So that's going to happen right here. The result is shown over here. You have a recombination of alleles. Alleles have been swapped between non-sister chromatids. So what that means is this. Let's look at our original orientation. There were just two combinations of alleles. You could have either a little d and a big E. So little d, big E. Let me write that more clearly. You could either have a little d and a big E, or over on this chromosome, you could have a big D and a little e. Big D and a little e. But since recombination has occurred, that is shown right here, what we have now is we still have the original combination, big D, little e, right? So big D, little e, same thing that we started with on the left. But we now have, on this chromosome here, we have big D, big E. This is a combination of alleles that did not exist before. And then right here, same thing. we've got a little d and a little e. Little d, little e. That's a combination that didn't exist in our original combinations of alleles here. You can only just have a little d and a big E, or a big D and a little e. Lastly, we have little d, little e, which is this chromosome right here. No crossing over occurred on this sister chromatid, so it remains unchanged. So recombination of alleles increases genetic variation. Increases genetic variation. Shown here again, crossing over leads to more variation in gametes. That's what, and that means basically, if you had 100,000 kids, not even 2 of them would be identical because all of this recombination of alleles. I'm going to skip through this one. I kind of realize I really already went over this. If you want to look at this slide more slowly on your own time, you may, but I really did just describe this. It's just a different diagrammatic version of what I was just talking about here. Here are the, yeah, okay, I'm going to move on. All right, metaphase 1. What happens in metaphase 1 is we have these tetrads, right? Got to use that word. That's what these are. This is a tetrad. And random orientation occurs, and that increases genetic variation, meaning the pink one could be on the left or it could be on the right. This math here, I'm not going to require you to know it, but there are 2 to the n possible orientations in metaphase 1 and also 2. That means that there's 2 to the 23rd in human beings, where n, remember, n equals the number of chromosomes, and there's 23 pairs. And if that math is intimidating to you, then you don't need to know it. Don't worry about it. Basically this, let's look at here. The pink one in example 1 is on the left, and then example 2, still on the left. But over here, it's on the right. Oops, let's call that example 3. And then over here, it is also on the right. The pink chromosome's on the right. Same thing down here. These are kind of different shades of brown. You could have them orient one of the browns on the left or the right, and that is going to lead to a different type of gamete, because the first division that's going to occur over here, you can see in metaphase 1, will be right down the equator of the cell, and the chromosomes will go left or right depending on what side of it they're on. And so whether the pink one's on the left or the right means, is it going to pair with this chromosome, or is it going to pair with this chromosome and produce the gamete here or here? This is an animation of what I was just talking about, and you can go to the PPT and open it up, but you can basically see right here that there's a whole bunch of different combinations of chromosomes based on random orientation. So here we could imagine the blue ones are the maternal chromosomes. So maybe here we've got 2 maternal chromosomes that are going to go this way, and then example number 5 here, you've just got 1 maternal chromosome that's going to the left. Example 6, you've got 0 maternal chromosomes going to the left. Example 2, all 3 maternal chromosomes are going to the left. You can access this hyperlink from the PPT, not from this video. So we've talked about prophase, metaphase. Now we're going to go over anaphase. This is pretty simple. Homologous pairs are separated and pulled to opposite poles. Note, we do not say sister chromatids, not sister chromatids. This is a chromosome bound to its sister chromatid. This is the reductive division, because the alleles that are on this chromosome on the right may not have those alleles. And so we say that the daughter cells here have a reduced amount of genetic information in them. Now you might say, but look, they still have 2 copies. But remember, these are sister chromatids, and the sister chromatids are not really 2 copies. It's kind of like, because they're 100% identical, it would be like having two copies of the same books. Yes, you could say you have two books, but you're definitely not going to make use of both of them because they're exactly the same. This, you don't need to mention that. I should have deleted that. I'm looking at microtubules. I'm not going to write that. So we've talked about prophase, metaphase, anaphase. Anaphase pulled the homologous chromosomes apart. The daughter cells are no longer diploid, and they each contain one pair of sister chromatids that are 100% identical to each other, except for where crossing over has occurred. You can see crossing over has occurred there, and then here as well, just so you can see. So telophase 1, not much to say. Interphase, there is no S phase in interphase 2, and that's because this is reductive division. If we copy all the DNA again, then we'd be back to where we started. Prophase 2, the nuclear membrane breaks down, no crossing over occurs. There is really not much to say here. It's really short. Metaphase 2, though, is a little different. Pairs of sister chromatids align at the equator. Spindle fibers form attached to the centromeres. Random orientation occurs again, and this does contribute to variation in the gametes, but only where crossing over has occurred. Because, for example, this chromosome, no crossing over occurred. So whether or not this chromatid is on the top or the bottom does not matter, because it is 100% the same. But the second chromosome here with its chromatid, notice crossing over has occurred at this end of the chromosome, so they are not 100% the same, but they are mostly the same. So crossing over does, random orientation does matter a little bit during metaphase 2, but not a lot. And just as an exercise, could you draw what metaphase 1 would look like for one of these cells? I'll do it for you. So if I was going to draw this chromosome with a big X and then this chromosome with a little x, what metaphase 1 would have looked like would have been two big X's forming a tetrad, and then two little x's forming a second tetrad. And then division would occur like so. But in metaphase 2, division occurs across the centromere. So the centromeres are actually going to divide. Thought we were getting to anaphase. Got a little ahead of myself. Metaphase 1 and 2 compared, which I just did for you. You can pause this and take a look at this if you're still a little confused. I'm going to move on. Anaphase 2. This is where the centromeres divide and the sister chromatids are pulled to opposite ends of the poles. Enough said. Telophase 2, nuclear membrane is going to reform around the sister chromatids. We're going to have this quickly followed by, if it is an animal cell, what was it called? A cleavage furrow will form, a contractile ring of proteins will form, and this will be pinched off into two cells. Or if it's a plant cell, they would just build another cell wall through the middle there. This is another link for a second, third, or fourth animation. If you want to take a look at it from the PPT, you should be able to extract it. How about this? Can you quickly, maybe take 2 minutes or so, you can pause the video, and can you explain what is going on in each of these phases to produce these 4 cells? Okay, and you can take a look at the where does crossing over occur, and it might also be useful to you to write a paragraph explaining all the stages of meiosis by memory. A couple of not exactly practice questions. These aren't examples of test questions or anything. They are more just concept check questions. You should be able to answer all of these. So check your language. This image shows what? Shows a tetrad. That is 2 homologous chromosomes paired up. They have synapsed. Check your language. This image shows 1 pair of sister chromatids, and they are joined by the centromere. They're 100% identical, except down here where apparently crossing over has occurred. How about this? This image shows 2 separate chromosomes. We know they're separate because they are different in size, and based on the coloration that is symbolizing banding and genes and alleles, they're different in size, and they also have different genes on them. What about this one? Two tetrads. So this is a pair of homologous chromosomes that have synapsed, and down here is also a pair of homologous chromosomes that have synapsed. What about this one? Which phase of meiosis is shown? This is C. It is metaphase 1, and here is the reason. You can pause the video and look at the reasons why we can identify this as metaphase 1. Which phase of meiosis is shown here, and why? This is going to be B, prophase 1, and the reason is shown down here below. Let me explain this in a little bit. The homologous pairs have associated, yes. So that's something that happens in prophase. Crossing over has taken place. That's something that also happens in prophase. This last one, homologous pairs have not aligned at the equator. Well, essentially what we're saying is that notice how they're not really lined up. So this would need to be oriented that way a little more, and then this would need to be bent over this way so that they form a nice, nice neat row like so. And like I said, this is just these are not practice quiz questions or anything. I'm just trying to get you to think about meiosis. Which phase of meiosis is shown here? So this is going to be A, interphase, and those are the reasons down there. I'll let you read through them, and I'm going to move on. This is a comparison of meiosis and mitosis, which is going to be relevant to the lab that we do. So here is the assignment. There are further directions above, more directions, and you'll have to get those directions from the Word document that I'm going to upload. So for now, if you'd like, you can pause the video and read the assignment directions. And I'm going to give you an example of how to start this. I normally don't do that because I can walk around the room and help you out. But since I can't, I'm going to help you get started myself by doing the first one. So the first diagram is a cell undergoing mitosis. That's not what we just talked about. We'll get to meiosis in just a second. You need to read through the directions here, but I'm just going to get going right away. So let me draw your attention to this. For mitosis, this cell, 2n equals 8, 2n, I've got a capital N there, equals 8. All right, so here we go. And also this, we're assuming that S phase has happened already. So S phase, check, it's already happened. So here we go. I'm going to draw a cell for you that is in prophase where 2n equals 8. There's a note for myself. 2n equals 8. S phase is complete. So that means that we should see sister chromatids. All right, so let's draw. I'm going to draw a pretty big cell, and you could probably get this done on one side, half a piece of paper, but paper is pretty cheap, so you might as well use a whole piece of paper, and that way you're not having to really try to fit things in there. So 2n equals 8. So let me just think about this, and I'm sort of modeling how I would think about it if I was a student. So that means that n equals 4. That also means that there are going to be 4 sets of homologous pairs. There's going to be 4 sets of homologous pairs. So I'm going to come up with a way of showing that. So let's say there's one chromosome, and there it is. So that is a result of DNA replication, right? It's a chromosome. That's one of the 8. One of the 8 S phase has occurred, so it's got its sister chromatid there. And then I'm going to draw another one over here. That's actually, you know what? I'm going to draw it a little bit differently. I'm going to draw it in a different color, and I'm going to draw it in blue. And I'm going to say that blue is we'll say paternal, and then we'll say, black is the maternal. Just to keep track of what I'm talking about here. So here is 1, 1, 2, 1, 2 chromosomes, we need a total of 8. So let's draw another one. I'll draw a little, let's say a little one over here. A little one, and then I'm going to draw the paternal side. I'll draw that one over here. So it's a little one. And all right, now I got to draw, let's say, so there's 1, 2, 3, 4, halfway done. Let's draw maybe a medium-sized one. So that's blue. So that looks like it's going to be paternal. I need a black one as well. And draw it here. So there's now it's 3, 4, 5, 6. And let's draw one more. Let's say we'll draw a monster chromosome just to distinguish them from each other, right? So there's the maternal, and then we'll draw another one like so. 7, 8. And you don't need to number them like this. I'm doing that because I'm doing this in front of you and I'm trying to keep my thoughts straight so I don't forget how many I've drawn. You don't need to number them like that. Then after this, you're going to be needing to label things as much as possible. So let's start with an easy one and label that as a centromere. Okay, I've labeled it once. That's good enough. You don't need to label every single one. That is simply excessively, I'm not sure what, too much, repetitive. There we go. So then I could label this, and then where was its homologous pair? I think it was right there. So that's a homologous pair of chromosomes. And let's see, another thing. I just realized that I didn't really... these chromosomes are all over the place, aren't they? So there needs to be a nuclear membrane here. Let's make a note here. Start in pencil. And I've written that in your directions here. So let's see, what's going on here? How could I fix this? Why don't I say... I'm going to draw a even bigger thing around here. There it is. Okay, so we'll call this, that's the cell membrane. That's the cell membrane, and this is the nucleus. Okay, better way to have done this would have been to draw it something like that, and then like so. But this is a PPT, and it's a little bit more difficult to draw here. So just make sure you label everything, and this, bottom line, is the types of type of diagram that I'd like you to do. 2n equals 8. Second diagram. This is the much harder one. The second diagram is a gonad cell undergoing meiosis. Once again, the 2n number is 8, and you will start out with a cell that will look just like the one starting in your previous diagram, mitosis. But this time, you will be representing cells which are undergoing the phases of prophase 1, metaphase 1, anaphase 1 of meiosis, and then again the same thing, prophase 2, metaphase 2, anaphase 2, telophase 2. I've said you can just skip telophase 1 because nothing really happens there. Use the same labels and annotations in appropriate areas of this diagram, and also label each cell as being haploid or diploid. So we'll start out with a diploid cell. Finally, include one crossover event between one pair of homologous chromosomes. You need to use colored pencils to color code each chromosome pair to explain independent assortment, or how we refer to that is random random orientation. Independent assortment is related to genetics, which we'll come back to later in another chapter. Next, use the same color coding for both diagrams. That is the mitosis diagram and the meiosis diagram. Remember that all chromosomes start as a connected pair of sister chromatids. You may use a book, PPT, for reference, but your diagram should be unique and not copied from any source. I doubt you'd be able to do that anyway. And the last one here. Grade is largely going to be based on colored, neatly drawn diagrams that are abundantly and appropriately labeled as I have directed. So let's begin here. So here's our cell. And you know what? I've just realized that, I generally don't require that you draw the cell membrane and the cell nucleus because it's sort of repetitive. So you can just use this a circle to represent the cell slash nuclear membrane. Sorry about that, membrane. And that's because, yes, in prophase 1 it's there and then disappears. We're not too interested in that. What we're really interested in here is just the behavior of the chromosomes. Okay, so let's begin here. There was a little one, then I recall there was a really big one as well, and then there was a medium-sized one, and then maybe, let's make that all bigger. Medium-sized one, and then maybe a second, maybe medium-sized one, except it's a little bigger. Okay, that's the paternal. And then we'll use a different color for maternal. What was it? It was blue and black. Oh shoot, I can't rewind in the videos. So anyway, here's they're the maternal and paternal, and then over here, same thing. Okay, and okay, I'm going to pause there. I'm going to wait to draw this last one. All right, so we have 1, 2, 3, 4, 5, 6, 7, and then I'll draw another one here in a moment, 8. So yes, 2n equals 8. Great. This is prophase. So I've kind of drawn the first one for you. This is prophase 1. This is what you're going to start off with. S phase has happened. Yeah, so that means that the DNA has been copied. We could label these things here as sister chromatids, sister chromatids. And then we could label these as a homologous pair. And we could also say, this is not big enough to label everything because I'm trying to draw it big enough so that you can see everything, but on a piece of paper you'd be able to do a better job labeling. See directions. You can also notice that synapsis has occurred. Synapsis occurs in prophase 1. And I've shown that synapsis has occurred in every single pair. The first pair, the second pair, the third pair, and then this fourth pair down there that I'm going to draw in a minute. You also need to draw one example of a crossover event, and I'm going to help you out with it. So let's say that this is one chromosome. It goes from here to there. And I'm going to say that there's, we'll just say a capital D representing some allele up there. And then I'm going to say there's a capital E down here. And on this chromosome, DNA replication has occurred. And, oops. Okay, so now there's the other, this is the sister chromatid. Sister chromatids are identical, so if on one arm there's a big D, there must be a big D here, and then there must also be a little e there. I'm using lines to just represent the loci of where these genes are. Alrighty then. Next, I said that we need to show one crossover event, and this gets a little bit messy. But there's our last chromosome here, and you can be a little funky with your drawing because it's got to be on the one hand clear but also biologically accurate. So there's sort of a compromise there. So here's the other one, and I'm going to show crossing over occurring. Whoa, that one's really long. So I'll just label this one crossing over. Crossing over. So I said that you need to show one crossover event, and I'm going to basically show you what prophase 1 would look like if we had a crossover event. I'm going to say that this chromosome here, so it's a homologous pair, so it has the same genes, but maybe different alleles. So I'm going to say there's just a little d, and then also here there's a little d. So same gene, different allele. It's the D gene, but this one's a big D and that one's a little d. So same gene, different allele. Down here, So there's the E gene, so it's got to be an E. I'm going to say it's going to be a little E. Sorry, try that again. Little. The software I'm using is a little cranky. Okay. Okay. It doesn't want me to draw an E there at all. Okay, there's a little E. Ha! Got it. And then over here, now it's getting a little messy. I know. Sorry. Doing my best. Little E. That doesn't like the E. Then there's a little E there. So what's going to happen is here, genetic information will be exchanged. Genetic info exchange. Ran out of space. Genetic info exchange. So this little e is going to get swapped onto the blue chromosome, and the big E from the blue chromosome will be swapped onto the red chromosome, crossing over. And if you're a little unclear as to what will happen next, then you need to refer to your book. It shows what happens. You can look at the my PPT as well. Remember, you should not try to copy parts of the PPT because it'll likely lead you to making mistakes. The point is not that you've memorized D's and E's on my PPT. The point is that you understand at a biological level what is going on here. So this is the first diagram of your meiosis diagram. This is prophase 1, as I've already stated. Okay. Well, that was a little messy. All right, so please don't hesitate to take pictures of your diagrams as you go and upload them onto Blackboard. I'm okay with you doing that. And you can ask questions about your diagrams if you feel that they are unclear. Don't worry about showing your work and having somebody else copy it. The point is that you use the creation of these diagrams to help you understand the material. That is our goal here. So feel free to be pretty open about what you're working on the discussion board. All right, hope this activity helps to clarify meiosis. It's a critical one, and it'll come back to us in another chapter.